Hello guys, so before getting started, I would like to thank Tess Brewer and Noah Fear for holding my hand all summer. And to the rest of the RECCS interns for creating such a supportive environment. You guys are awesome. So, the last 8 weeks we have been looking into bacteria that possess the ability to solubilize aluminum phosphate. So later the details of the experiment will be discussed, but first I'd like to share with you the reasoning behind all of our efforts. So, Okay. So tropical forests are primary carbon sinks in the world, and because of this, a strong understanding of our nutrient cycles within that climate and the limitations these nutrients may have is crucial to furthering our knowledge of natural carbon sequestration. One of the main ingredients vital to a healthy forest is phosphorus. Phosphorus is needed for protein synthesis, the development of new tissue. It's pretty much used in every metabolic process. We really like to add it to our fertilizers to maximize plant growth, and is completely essential for all forms of life. So the main issue, or the most interesting thing about phosphorus is phosphorus throughout the world is mainly biologically unavailable. So this map illustrates the total distribution of phosphorus throughout the world's soils, and as you can see, almost everywhere is on the lower end of the spectrum. Now, phosphorus is an extremely reactive element, and because of this, it's never found on Earth in a free state. In fairly unweathered newer soils, phosphorus tends to bind with calcium and magnesium, whereas in tropical highly weathered areas, your phosphorus tends to bind with aluminum and iron, resulting in an insoluble and therefore biologically unavailable compound. Now, given that a large portion of the biodiversity hotspots are located within these tropical climates, further investigation into insoluble forms of phosphorus needs to be done to gain a better familiarity of how phosphorus is mobilizing throughout tropical soils. Now, there are a multitude of factors playing into the phosphorus cycle. As you can see here, the phosphorus cycle is actually a slowest moving nutrient cycle, with most of our phosphates coming from the weathering of rocks. Now, as mentioned before, this experiment was focused on the role bacteria play in phosphate solubility, so it's going to be this area right here. And it was also designed with three main questions in mind. What kind of bacteria communities are present in this soil type? Out of these, can they solubilize aluminum phosphate? And if they can, do they also have a tolerance to aluminum toxicity? So 8 samples were used in this experiment. 5 of our tropical samples were from Panama and 3 were from Montana for a contrast in soil climate types. The soil inoculants were placed on 3 defined media types. We had a plate with no phosphate for our negative control, a plate with potassium and sodium phosphate for our positive control, and a plate with aluminum phosphate for the experimental aspect. Now, these plates were left for an 11-day incubation period before DNA was harvested and purified, and then sequenced using the 16S ribosomal RNA amplicon method. So shown in this graph are the colony forming units found per gram of soil for each plate type. Each individual graph is each soil sample. Now, as a side note, 1 teaspoon of soil can contain up to 1 billion bacterial cells, which is why our numbers on our y-axis are so large. Our x-axis is our 3 defined media. And as you can see, our positive control, which is shown in green, has the highest growth. But what was interesting is there was more diversity on our negative control, which is our phosphate-free plate shown in red, and our experimental, which was shown in blue, which are our aluminum plates. Now, there are more than two reasons why this could be happening. There's more competition on our control plates, which is going to limit the diversity of the plate, and there could be more bacteria than originally expected that can live in low phosphate conditions. Now, this figure shows the top 6 taxa that were found with the noticeably higher abundance on our aluminum plates compared to our positive control. Now, these are the taxa that are most likely to have an aluminum resistance. However, more testing needs to be done before arriving to a definite answer. Now, one of the more interesting of these is the genus Bradyrhizobium, which is this guy here. Because many bacteria within this genus can also fix nitrogen. Now, the other 5 genus have little to no ecological research done on them, with most research I could find pertaining to human health. So our next steps would be to take the top 6 taxa that were found and administer a toxicity test to see if these bacteria can actually tolerate being in an environment with high levels of aluminum. I think an attempt to make a more pure media with aluminum phosphate as the only source of phosphate should also be done to see if these bacteria are actually solubilizing aluminum phosphate, not just living on the trace amounts of phosphate that may be present. And more research should be done to see if the other 5 taxa that were found play any ecological role whatsoever, and a broader sampling of tropical soils should be added to this to gain a solid foundation of the bacterial abundances in these soil types. So out of these 3 questions the experiment was designed around, I'm happy to report we have an answer to the first one. We know who's present in the soil of the 2 sites we tested. We can use that information to proceed to the next steps. However, this was just a really small piece in a large-scale project. I still deem this a success, and I'm excited to see what other data microbiologists and soil scientists can bring to this project. Here's my reference.